We'd like to thank you for joining us for the Kansas 4-H Equine Science webinar series for the month of March 2023. I'm Kelsey Nordyke, your 4-H Ag Science Specialist, and joining me today is Dr. Jenny Borman. Dr. Borman is an Assistant Dean in the College of Agriculture with K-State and also a professor in the Department of Animal Science. Today, Dr. Borman is going to share with us about equine genetics. And before we get started with that, I do want to remind our Kansas 4-H members that there have been some changes to the qualifying process for the Kansas State Fair 4-H Horse Show. Be sure to check out the Horse Project page for information on that. And if you have any questions, don't hesitate to reach out to me. And with that, I'm going to go ahead and turn it over to Dr. Borman.
All right, thanks so much, Kelsey. Okay, so you should be able to see my screen now. And what I'm going to talk about today is just an introduction to equine genetics and specifically equine color genetics. This is something that I've taught for a lot of years here at K-State. And genetics is a tricky subject and it's difficult for a lot of students, but we're going to try and sort of boil it down to kind of the most basics and start to understand what makes our horses the color that they are. And so again, it's a hard subject. And so if you don't understand all of it, that's okay. It's something that you can, go back and look at again and hopefully understand it as you go through your science courses. So first of all, so just some basic genetics definitions. There we go. So when we think about a cell, the inside of the cell has structures called chromosomes, and those are just like little threads, little linear things. And on those chromosomes are what's called genes. And the genes are what contains the information that makes something happen in the body. Okay, and these genes are passed on from parent to offspring. And so those are really the vehicle by which information gets transmitted from generation to generation. And then that information causes something to happen. It makes that horse gray or not, or whatever it is we're talking about. Now, every gene has multiple forms that it might take, and we call those alleles. Okay, and so generally we're just going to use letters to designate alleles, so capital or small letters. Sometimes we might use something like 2 letters or a letter or a number or superscripts. There's a lot of freedom on how we're going to designate that, but some combination of letters are going to indicate the allele. Now, every animal has 2 chromosomes. A pair for every single set of chromosomes. And so every animal is going to have 2 alleles, exactly 2. And when we want to write out what we call the genotype of that animal, that's going to be the 2 letters or the 2 alleles that each animal has. So the genotype is the 2 letters. The phenotype, that's what we see. So bay is a phenotype. The genotype for bay is some letters that we'll learn about. So it's important to keep genotype and phenotype straight. Now, animals could have the same 2 letters, the same alleles at a gene, or they might have 2 different ones. And so this figure is just showing a pair of chromosomes. And so for the R gene, whatever R is, this particular animal is what we call heterozygote because they've got a capital R on one side and a little r on the other. Okay, they've got the 2 different R alleles. An animal might have 2 of the same. So if this animal had 2 big R's, we would call that homozygous. Okay, so homozygous means they have the same, maybe 2 bigs, maybe 2 littles, doesn't matter, 2 of the same. The heterozygote means they've got 2 different alleles. Okay, so when we think about how those two alleles can work together at that gene, we have a couple different ways they might interact. Probably the most common one that we're most familiar with is what we call complete dominance. And that's where one of those alleles will just completely cover up the other one. We don't even know the other allele is there because one of them dominates. We call that the dominant allele. And just by convention, we're going to write that one with a capital letter. The dominant allele gets the capital letter. The recessive allele is the one that gets covered up that we cannot see. We write that with a small lowercase letter. Okay, so kind of a classic example that we use when we teach ag genetics all the time is Angus cattle, coat color. They can be red or they can be black, and that's controlled by one gene. And so in the case of this particular gene in Angus cattle, the black allele, the black coat color, is dominant. So we write that with the big B. And then the red allele is the recessive one. So we write that with a little b. We use the same letter because they're at the same gene. And we just use capital for dominant and lowercase for recessive. And so every animal, every cow in this case, is going to have two B's. They might have two big B's, they might have two little B's, or they might have one of each. Okay, two different homozygotes and a heterozygote. Now, the reason that we only have the two phenotypes, black or red, is that the big B is completely dominant. So if you've got a heterozygote animal that has one of each, then they're going to be black. So when you look at a black Angus bull or cow, you cannot tell by looking at that black Angus whether they're big B or big B little b. So that's a difficulty that we have when we're dealing with completely dominant traits, is that we can't tell by looking at them what the genotype is. Now, if she's red, that heifer cow is red, then that's easy. We know she has to be little b. Okay, so that's the way that many of the genes that influence horse coat color work, complete dominance. However, there is another type of gene action that we see in horse color called incomplete dominance. And that's where one allele doesn't cover up the other. The heterozygote is kind of in between. And so our classic example would be on actually not just snapdragons, but several different kinds of flowers, where the homozygote is maybe red, the other homozygote is white, and the heterozygote is pink. So kind of a blend or a mix. Now, in the case of incomplete dominance, we are going to write a capital and a small, because if one allele doesn't dominate the other, it's not really proper to write upper and lowercase. And so we're just going to use superscripts and call them R1 and R2. So in this case, if R1 is red, R2 is white, the homozygote for R1 is red, the homozygote for R2 is white, and then our heterozygote is in between. So those are our pink flowers. And we're going to see both of those different types of gene action when we talk about horse color. Okay, so just some general things to think about is that people defined horse colors hundreds of years ago, before we knew anything about genetics. And so the common names that we use for colors don't necessarily match up with the genes. Sometimes you might have one genotype, and we may use 2 or 3 different words to describe that color. Or vice versa. You might have one genotype that has more than one color. So doesn't always line up one-to-one. And a really good example of that is chestnut and sorrel. Okay, so some breeds will let you register chestnut and sorrel horses differently, but the genes are not different at all. Chestnut and sorrel are exactly the same thing. And so there's no genetic difference in any way. That one's more of just almost a discipline thing. Chestnut, like if you're doing thoroughbreds or Arabians or some of those kinds of breeds, you would use the word chestnut. Stock breeds tend to use the term sorrel, although quarter horse has both chestnut and sorrel, but it's not really any different. The other thing that we run into is that there are some combinations that don't have a name. And we'll do some examples of those later on. So you may have a certain genetic combination that results in a color, and there's not even a name for that. So when you register that horse, you pick something that's close and is not right. And again, we'll look at some examples. Another thing to keep in mind is that every horse has a color. They may have lots of white markings. They may have a paint or pinto pattern where they're predominantly white, but they still have a base color. And so we're going to kind of ignore the white markings part of it and focus on the base color for now. Okay, so we're going to go through some of the different colors in the genes. I don't have pictures, so kind of copyright issues. And so what you could do is maybe pause this for a second, open another window, and then just use your favorite search engine to search horse color white. And it'll bring up all kinds of pictures. So if you'd like to do that to kind of look at the pictures, if you're not familiar with the coat colors, you can do that as we go along. But I think white is an interesting one because when I grew up, I was always told, or you read in the books, that there's no such thing as a white horse. That what you're thinking is a white horse is really a gray that's old enough that he's turned white. And that's true probably 99 point something percent of the time. Most of the white appearing horses that we see are actually gray. However, there is such a thing as a white gene that causes a white horse. Now, it's very rare. We don't see very many of them at all, but it is present in several different breeds, including thoroughbreds and Arabians. And so again, go to your favorite search engine and search white thoroughbred, and you'll see pictures of horses racing that are literally pure snow white. And so white is a dominant allele. And so we designate the white allele as big W. The little w, and I know W's are hard to sometimes tell capital and lowercase, but the little w is then not white. So that would be like every other horse on the planet that's not white would have the two little w's. Now, what's interesting about white is that if the horse has two big W's, it does not survive. That's lethal very early in embryonic development. One of those lethals that you breed the mare, 21 days later, she's back in heat, and you think, oh, she didn't settle. Well, she might have had a conception, but if it was big W, that conception died within a few days. Okay, so that's a homozygote lethal. So if we look at the different combinations of genotypes we might have, big W, those are dead. We never see those. They die in the first few days of pregnancy. The heterozygote is the horse that's white. And again, if they're little w, that's like every other horse on the planet. 99.999% of horses on the planet are little w. But what's also interesting about white is it sort of trumps every other color gene. No matter what genes your horse has for color, if they've got a big W, they're white. It covers up everything. Okay, so that one's interesting. Very rare, but interesting. The next one we're going to look at is gray. And this one is much more common, right? We've all seen gray horses. Gray is interesting because it has a wide variation of expression, and it's progressive, right? We know that most foals that are going to be gray they're born bay or sorrel or black or whatever. They're born some other color where they shed out and they become gray. And they usually are very dark gray as younger horses, and every year they get a little bit lighter. Gray is also caused by a dominant allele, and so we're going to designate that one as big G. Big G causes gray. Little g then is the not gray allele. Now, unlike white, G is not lethal. So if you have a gray horse, just like our Black Angus, that gray horse might be homozygous for big G, or they might be heterozygous, and you can't tell by looking at it. But every other horse on the planet that's not gray is going to be little g at the gray locus. Gray is like the second highest trump card in your deck, because other than W, gray covers up everything else. So if your horse has a big G, doesn't matter what other color genes they have that we're going to talk about in the next few minutes, gray is going to cover them all up. And so you don't know what those horses have for color genes other than at least one big G. Okay, so white and gray, those are kind of the trump cards. They cover up everything else. But probably the thing that's most important to understand is this interplay of chestnut or sorrel. I'm going to use the term chestnut, but if you want to use sorrel, that's fine. But the interplay of chestnut, bay, and black. Because if you look at most breeds of horse, these three colors would, actually chestnut and bay really, would probably catch like, what, 80, 90+% of horses. By far the most common coat colors in most breeds. And we generally call these kind of the base colors. But what's interesting about chestnut, bay, and black is it's not just one gene that controls this. There are two genes that work together, sometimes cover each other up, to determine whether that horse is chestnut, bay, or black. And that's, again, assuming that they're not gray. And so those two genes that we're going to be looking at are called extension and agouti. They were named based on other species before they were discovered in horses, and so that's why those names kind of seem weird. But we got to understand how those two genes work together. Okay, so first of all, we're going to look at extension, E. And E is almost like a switch that determines whether that horse can produce black pigment or not. Okay, so when I'm talking about black pigment, I'm just talking about any black hair anywhere on the body. Okay, if this horse has a big E, okay, the big E is the allele that allows the body to create black pigment. So if the horse has a big E, he won't be chestnut. He's going to have black pigment somewhere on his body. We'll figure out where in a minute, but he'll have black pigment somewhere. If this horse has two little E's, the little E says no black pigment. Okay, so if this horse has two little E's, his body is unable to create black hairs, unable to create black pigment. He's missing an enzyme or a catalyst or something that would allow that to happen. So any horse that is little e is going to be chestnut, no matter what that A gene that we're going to look at here in a minute says. Doesn't matter. Little e cannot make black pigment. Now, if they're big E, they're going to not be chestnut. We don't know yet what they're going to be. We've got to look at our agouti gene here in a minute. But we know if they've got at least one copy of big E, their body will produce black hair. Okay, so little e, no black hairs, we're going to be chestnut. A big E says, okay, well, we're going to have some black hairs. We got to figure out where. And that's where agouti comes in. Agouti is what controls basically where the black hairs go. And again, it was named after, I think, a mouse or a rat or something. The agouti is where that name came from. But what agouti tells us is that the dominant allele, the capital A, only allows black hairs on the legs, mane and tail, tips of the ears, the points. Okay, well, if a horse only has black on his points, that makes him a bay, right? That's the definition of bay. The recessive allele, the little a, allows black hairs throughout the body. So that would result in a black horse. And so you can see here that if they have a big E, they have to have a big E at extension in order to make black hairs at all. But assuming they've got a big E, then if they've either homozygous or heterozygous with a big A, that's going to make them a bay horse. And only the little a, the homozygote for little a, will be black. So you always have to look at E and A together to determine first, chestnut or not chestnut. If not chestnut, then bay or black. And so I put this table in here for your reference where you can look and say, okay, if I look at all the combinations of both extension and agouti, then what is my phenotype? So again, if you look at these first 6 rows, they've all got at least one big E, which means we will make black pigment. And so then we look at A, big or big little a is bay, little is black. But then these bottom 3 rows show that if they're little e, it doesn't matter at all what's at A, because those are going to be chestnut horses. They are incapable of making black pigment. Okay, so if you remember one thing about horse genetics that will be useful, it's probably this. It's E and A, because that's going to control almost all the horses that we see. Now, because we might want to know those things, we actually have gene tests. And so if you look through like breed magazines and look at stallion advertisements, you might see advertised some of the results of these gene tests. So for example, if you have a bay horse, you don't know for sure if he's big E or big E little e. And you might want to know, in a lot of our disciplines, a bay or a black horse, a dark colored horse is preferred to a sorrel horse. And so you will see advertisements that say things like, does not carry the red gene, he will not sire a red foal, homozygous for black factor. So I wrote some of them on here. What they're saying then is that horse is only going to be able to sire bay or black. It's a little bit misleading because when they're talking about extension, they talk about red factor and black factor. Well, a horse that's homozygous for big E, for black factor, he's still going to sire bays, right? And so saying black is a little bit misleading if you don't understand how E and A work. What they're really talking about is extension, and he won't sire a red horse because all of his offspring, if he's homozygous for big E, will be either bay or black. They will have some black pigment. Now, you would never actually test a horse that was little e, because you know by looking at them that they are chestnut and they have to be little e. It's the bays and blacks that you want to test to figure out if they're a carrier of little e or not. You can also test for agouti if you want to see if your horse is homozygous bay or heterozygous, in which case they would carry that little a allele. Now, I put the gene test on here because this is some of the ones we commonly see, but you can actually get gene tests for almost any color gene that you're interested in, if you want to do that. Okay, so a question we often hear is, well, what about brown? Well, most breeds have a brown category that you can register your horse. And we know that brown is a very, very dark horse. They pretty much look black, but they've got a brown nose, brown elbows, brown flanks. Well, it turns out this is one of those times that genetics and what we call things doesn't line up, because there's no such thing genetically as brown. There's not a brown gene. What we think is that most brown horses, and genotyping gene tests confirm this, they're really just dark bays. There are some other minor modifying genes that might, for example, if a horse was black at E and A, there's some modifiers that might make that brown be a little faded. There's some things we don't know yet about, but we do know that there's not necessarily a brown gene. Now, there is another species. There's a brown that's very common in other species. Think chocolate lab, that kind of brown. And if you think about a chocolate lab, we really don't have horses that color, right? Because that is not a gene that is important in horse color. All horses are completely homozygous. There's no variation. We just don't have that brown gene that they do in lots of other species. Okay, so now we're going to talk about some of the dilute colors. The first one we're going to look at is dun. And what dun does, the dun gene, it dilutes the color, so it makes the overall color lighter. But dun also causes what we call primitive marks, which is a dorsal stripe down the back, sometimes barring around the legs, zebra stripes on the legs, or across, a stripe across the withers. Again, go to your favorite search engine, type in dun, and look at some of those pictures. And we often call this allele the dun factor. You hear people say, oh, this horse has a lot of dun factor, which just means that they were done with lots of primitive marks. But this is also a dominant allele. So big D is going to be our dun allele, and little d is not dun. So that's kind of what every other horse has. Okay, so if they're homozygous or heterozygous, they'll be dun. And if they've got two little d's, they're bay or sorrel or something else. And so if we think about what that does to the base color, if a horse has dun factor on bay, that makes them a zebra dun. If they have dun factor on chestnut, they become a red dun. Dun factor on black makes what we call a gruya. Another dilution that is relatively common is called cream. Now, this is one that is not completely dominant. So remember we talked about the flowers that are red, pink, and white? Well, that's how cream works. And so I'm going to designate those alleles with superscripts. And so the cream allele, I'm going to call C with a little superscript CR for cream. And then the not cream is going to be a plus. Okay, so most horses, bays, black, sorrels, whatever, they're C+, just regular, quote, normal color. If they are heterozygous, they have one maybe dose of dilution, so the coat will be lighter. If they have two alleles for cream, they're going to be even much more lighter. So like 2 creamers in your coffee, for example. So what does that mean? Let's look at what that is. So again, C+ horses, they're bay, chestnut, black, whatever. They're regular dark coat color. If they're heterozygous at cream, and they have bay genes besides that, they're going to be buckskin. If they're heterozygous cream and they're chestnut, that makes palomino. And heterozygote cream on black makes what's called smoky black. Now, smoky black is sometimes just looks black, but sometimes it's a little bit faded. And so this would be one way you might get a quote brown horse, a slightly faded black. If we've got homozygous cream, so 2 copies of the cream allele, these are called double dilutes. And in this case, the bay horse would be Perlino, which is basically white, sometimes a little bit of orange in the mane and tail. Chestnut becomes cremello, and black becomes what's called smoky cream, which basically looks the same as cremello, or sorry, same as perlino. But all three of those are so light that you can't necessarily tell by looking at them, oh, that's a perlino, oh, that's a cremello. Sometimes you can, but not necessarily. Again, go to your search engine and search up these colors if you want to see some of the differences. Okay, the third modifier we're looking at is roan. And roan is just a scattering of white hairs across the body. So kind of a frosting almost. But in general, the head stays dark and the legs will stay dark. Roan is also, like dun, a dominant allele. So if a horse is homozygous or heterozygous for the from R, big R, they're going to be roan. And if they are little r, they'll be bay or chestnut or whatever. They're not roan. Now, this is one where, depending on what breed or discipline that you work in, it might be a little bit tricky. So what I have kind of here on top is our stock breed nomenclature. So if you're looking at quarter horses or paints or something, if you put roan on bay, we call that bay roan. Makes sense. If you put roan on chestnut, we call that red roan, and roan on black becomes a blue roan. However, if you go to maybe Arabians or some of the other non-stock breeds, they use the term strawberry roan. So you've probably heard that. Strawberry roan for the chestnut base and red roan for the bay base. So that can be really confusing because we both use red roan, but you use them for two different things. So make sure you kind of are clear about what breed or who you're talking to, whether you're talking about a bay base or a chestnut base. Now, those are the main ones, and we catch 98% of most of our breeds. But there are some other rare color genes, things like silver dapple or champagne. There's just a few other things out there that are relatively rare. But also, we could have combinations of things that don't have an actual category. For example, what if you had a chestnut horse, but he had cream, so he should be palomino, but he also has dun, so he should be red dun. But maybe he's roan also. I guess that's a roan dunlino. There's not a checkbox for that when you register a horse. So you would misclassify that horse as something else. So just kind of for your reference, I went through and looked at sort of our common colors and then wrote out the genotypes that define those. So I guess I didn't talk about this. Whenever we use a dash, that means we don't know what the other allele is. And actually, this is, oops, that's a mistake because we do know on that one that this other allele must be a little W, because if it was two big W's, he'd be dead. But for example, a gray horse has to be two little W's and then a big G, and we know nothing else. Now, for our main kind of base colors, chestnut, bay, and black, we know they have to be not white, not gray. If they've got two little E's, they're chestnut. We don't know what their A's are. That's what the dash right there means. We don't know what their A's are. But if they're chestnut, we know they're not dun, not cream, and not roan. Okay, bay is going to have, again, not white, not gray. It's going to have at least one big E and at least one big A, but we don't know for sure what the other one is. Could be big, could be big, little. But we know for sure they are not dun, not cream, and not roan. And then similarly for black, just the difference is they're little a, little a. Now, if we look at our dun grouping, the bay, chestnut, black, that's copied down here exactly, except now instead of little d, to be a dun, they've got to have a big D. Maybe two big D's, maybe big D, little d, we don't know for sure, but they're still not cream and not roan. Okay, if we look at our cream variants, okay, again, not white, not gray. The buckskin and the perlino will have the bay base, so big E, one big E and one big A at least. The palomino and the cremello have the chestnut base, so two little E's. We don't know about A. They're not dun, they're not roan. The buckskin and the palomino are heterozygous for cream. The perlino and the cremello are homozygous for cream. And then the same kind of setup for our roans. Not white, not gray. The bay, big E, big A. the blue, big E, 2 little a's, and the red, 2 little e's, we don't know what's a day, not done, not cream, and then either homozygous or heterozygous for roan. So that's one kind of a quick reference so you can see what are the genotypes of colors that we would commonly define. Now, the other way is we might look at a genotype and try to figure out what color that horse is. So if I give you this genotype and say, well, if you had this, what is this horse? What color does he look like? Well, you just start at your W's and you look at every gene independently. So little w says not white. Little g says not gray. Big E says not chestnut. Okay, the big A says bay. So far we know he's bay. But he has a dun factor. So he's bay and dun. Not cream, not roan. So overall, the final answer for this horse would be our bay or zebra dun. Okay, let's do another one. Not white, not gray. Big E's tell us, or a big E tells us he's not chestnut. Little a says he's black. So far we have a black horse. He does not have dun factor. He does not have cream, but he does have roan. So a black base with a roan allele is going to be a blue roan. Okay, the third one, not white. This one's gray. He's heterozygous for gray, has one big G, so he's gray. And now we're done. Because remember, gray covers up everything else. So it doesn't matter what that horse has for E or A or cream or any other gene. Gray is going to cover that up. And in fact, the horse that my daughter shows, Palomino horse, both of her sire and dam, both of her parents were gray. Obviously, maybe it was one of these. One of those must have carried the cream because she's a Palomino, but couldn't tell because that was completely covered up in that horse. Okay, last one. Not white, not gray. Little E's say chestnut. So if they're chestnut, remember A doesn't matter. Doesn't matter that he's got bay and his agouti gene. Don't worry about that one. He's not done. He is cream. So we've got chestnut and cream so far, not roan, and this would be your Palomino. Okay, so that's how you go through and figure out if you've got a genotype, what that color might be. Okay, we're gonna take a few minutes now and talk about patterns because many of us might have breeds where they've got a white pattern besides a base color. So we'll start out talking about paint or pinto. I'm going to use those interchangeably for this. And generally we talk about Tobianos, Overos, and Toveros. But again, people decided what those meant a long time before we knew anything about genes. So they don't necessarily match up with what we know now about genetics. But in general, the way these were defined was that Tobiano usually had dark on his face, usually had white legs, often had white crossing over the top line. What's interesting is that a very minimally marked Tobiano might be pretty much just a dark horse with 4 high white socks. A maximum Tobiano might be almost all white. So the amount of white and the amount of dark can vary greatly within a color category. Okay, Overos, I use the word Oreo to remember Overo. They generally are dark along their top line. They're dark on their legs and have white in the middle of their body. And some horses combine kind of both sets of characteristics and they didn't even think of a new word. They just combined those words to call those Toveros. Now, within those 3 categories, we actually know now there's way more genes going on and way more different patterns than that. We didn't know that when they first defined what a pattern was. So now we have things like Sabino and splashed white and dominant white, all controlled by different genes. Tobiano is its own thing. We'll talk about that in a second. But all these other patterns usually just kind of get lumped into Overo or Tovero. Another complication is that a horse could have more than one of these. A horse could be a Tobiano and a Sabino, or a splashed white and a frame. And so we might have lots of different pattern things all going on at the same time. Okay, so Tobiano, that's kind of the simple one. It's actually one single gene that controls the Tobiano pattern. And it's a dominant gene, dominant allele. So big T is Tobiano. So a horse that's homozygous or heterozygous is going to have the Tobiano pattern. And then all other horses in the world, all non-Tobianos, are little t at that gene. Now, you could argue that it's not quite a completely dominant gene because in general, there is, on average, you can sort of have a prediction whether it's homozygous or heterozygous. So the homozygous big T, they tend to have what's called paw prints or kind of the roning areas on their white, shadows, I guess. Definitely not absolutely a rule, but that's an indication that they may be homozygous T. What is not is the amount of white. You know, it sort of makes sense. You might think, well, if they're big T, they'll have a lot more white than if they're heterozygous, and that is not true at all. A maximal or minimally marked Tobiano might be big or big little. There doesn't seem to be a pattern. And something else that makes it complex is that, and I say it this way, the genes, biology didn't read the rule book, right? People decided that this is what makes a horse a paint or a pinto. When we made a rule book and we drew lines on a picture of what that horse has to have white to be considered a paint. Well, biology and the genes and the chromosomes, they don't read the rule book. And so they don't necessarily match up exactly with what the genes do and what the rule book says. So you might have a horse with a Tobiano allele, and he might just have a lot of white on his legs, but he doesn't quite fit the mold or qualify for what we decided years ago was the rule to be a Tobiano. So again, it can be very confusing because these things don't always just match up the way that we think they should. Okay, the most common of the Overo patterns is called frame. This one is also the lethal white Overo. Okay, so you might have heard of this one. And this is sort of the classic Overo pattern where they're dark on the top, dark legs, white in the sides. And this frame allele is a lethal in the homozygous state. So very similar to the white allele we talked about way at the beginning. And so if a horse is homozygous for frame, big F, that foal will be born and it'll be white, and then it will die within a few hours. It has an intestinal problem. So the horses that you see, that are frame Overo, are actually heterozygote. Every frame Overo that you see is a heterozygote, big F, little f. And then every other non-frame horse in the world is just little f, little f. So this one, it took them a long time to figure out because they thought it was recessive. Because Overo is one of those that a lot of horses that are heterozygous, so they're big F, little f, they have frame, but frame didn't read the rule book either. Frame doesn't know that the white has to extend beyond a line drawn around the middle of the knee or whatever the rules say. It didn't necessarily, frame doesn't know that. And so there's there's horses out there that have the frame allele, but are not registered as paints or pintos. They're just regular old quarter horses or whatever, thoroughbreds or whatever, because they don't have quite enough white to qualify. And because of that, we occasionally will see a lethal white horse born when you mate an Overo to what is just a dark, regular colored horse. Well, that horse maybe had, or it did, if it had a lethal white offspring, it had frame, it just didn't show it. And so that can make lethal white a little bit tricky. And so not a bad idea to test, even if you don't think your horse is an Overo. And so there is a gene test. And a lot of times you actually see it abbreviated OLWS, Overo Lethal White Syndrome. And they'll say they're either not. So NN means no lethal white. So that'd be like my little f. You in theory would never see OO because that would be the foal that died. If you took a DNA sample from the foal that died, he would be OO. And then the normal Overo horse would be the NO. So not a bad idea to test if you have any idea that that horse might have a frame allele. Okay, some other ones that are a little more recently discovered is Sabino, is one that we often see with the roning. So it's a paint that has lots of speckles and roning on it. Sometimes they can be almost solid, just a big blaze and white legs and maybe a little bit of roning on the edges, and some will be almost pretty much almost entirely white with maybe a little bit like a dark, like a bonnet, a dark spot on their head. So quite a range in the phenotypes that we see with Sabino. And the Sabino gene, we're going to label it SB1. So 3 different characters for one gene. The capital then is the Sabino. The lowercase is the regular non-Sabino. And I put mostly dominant and mostly recessive. It's a little bit incompletely dominant because the homozygote capital S's are the ones that are really, really white, almost entirely white. The heterozygote, one copy of Sabino, one copy of non-Sabino, they tend to be sort of the classic Sabinos, lots of ronin, lots of white markings, but their whole body isn't white. And then most other horses in the world are just little less, they're just not Sabino. Splashed white is a rare pattern. We don't have many of these, at least here in North America, but it's characterized by white on the belly, white on the legs, and maybe white on the face. It got the name because it almost looks like they ran through paint because a lot of the white is on the lower part of their body. Again, it can be variable with the amount of white versus the amount of dark. A lot of times these horses have blue eyes, not 100%. And it turns out there's actually 6 different alleles that cause splashed white that we know about so far. 10 years ago, I think we only knew about one of them. So we're discovering more of these all the time. And so they're completely different alleles that can all cause splashed white. And so I kind of wrote it with just a number sign here because it could be splashed white 1, it could be splashed white 3, whichever number it is, has sort of the same action. One thing that we don't know for sure, we think that some of these are lethal in the homozygous state. So your big S animal will either be splashed white, I think it's 3 and 5 maybe, that potentially are lethal. We just have not found any homozygotes yet. So maybe homozygote lethal. The heterozygote big S little s for all of these will be splashed white. And then our our solid colored horses, non-paint, non-pinto horses are all little less for all 6 of those alleles. And then the most recent one that's been discovered probably in the last 5 years or so is what we call dominant white. And these have a huge range of expression. Sometimes they're actually almost completely white with maybe a few dark hairs on their top line. And some of them almost appear solid with just some white, high socks, for example. And we've so far found 5 of those. I'm not sure why they're named the numbers that they are, but we've got 5 different dominant whites that have slightly different phenotypes. But like splash, some of these are homozygote lethal. So if it's a certain W and they've got big, they're going to be lethal. If they're heterozygote, they're going to have whatever that spotting pattern is for that gene. And then most horses in the world are simply little w for those alleles. Okay, so a lot going on in paints and pintos, and we don't know all of it. Like I said, a lot of these have been discovered just in the last 5 years. And so there's probably more. There's probably more of these white spotting alleles that we just haven't discovered yet. Just a quick note, you might have heard of something called a crop out. And that's when, for example, 2 quarter horses or 2 thoroughbreds or 2 solid colored horses will have an offspring that has a paint pattern. And probably this is because one of those parents had frame, or it had splash, or it had Sabino, it had one of those alleles, but was minimally marked and didn't get registered as a paint. Again, the genes don't read the rule book. They don't know where the white is supposed to be. But occasionally we have new mutations, and all these W's that we found have been new genes, new mutations that arose from solid non-patterned horses, and now there's a mutation, and now we have a new spotting pattern. But it's important to note that even a horse that's minimally marked, it can produce spotted paint pinto horses just like any other. So that can make breeding these a little bit tricky. You can't quite tell what the genotype is from the phenotype. Okay, the last pattern that we'll talk about is leopard. And so this is something that in our criteria, we think about this primarily with the Appaloosa breed or maybe the Pony of the Americas, but there are some other breeds around the world that also have leopard. Okay, so it's not just an Appaloosa thing. What's really weird about the Appaloosa pattern, the leopard pattern, is that there's lots of different sub patterns. And if we have any Appaloosa people, you know what I'm talking about. Things like the frost or the blanket or the speckled or the varnish. There's lots of these different kinds of Appaloosa patterns. What's weird is they're all triggered by this one gene, the leopard gene. Now we think there's a lot of different subgenes or modifier genes. We found one. We know pattern 1 is a modifier, and there's probably others that we haven't discovered yet, but they're all triggered by the main gene that we call leopard. And so we designate that, again, it's a dominant allele, so we designate that with the L, big L. And then all other horses that are not leopard are homozygous for little l. Generally, I put dominant recessive. It's a little bit incompletely dominant because generally the homozygotes tend to have more white on them than the heterozygotes. But again, that also is going to depend on those modifier genes that work with leopard. The other thing we have going on with the leopard pattern is that these homozygote horses for leopard generally are night blind as well. And so that's a problem that we see in the homozygotes. Okay, well, that was a lot of genetics in a very short amount of time. And so hopefully at least got an introduction to how some of the base colors work and what some of these modifiers are. So again, I know it's a lot of stuff, but I would be absolutely happy to answer any questions that you might have later. Just shoot me an email, pick up the phone, and we can go into more detail, or I can explain what some of these other genes are. And for sure, as you get into your genetics classes in high school and eventually college, hopefully this stuff will make that a little bit easier as we get there. So I'd be happy to answer any questions, and I'll turn it back over to you, Kelsey.
Awesome. Well, thank you, Dr. Borman. And we're going to leave this slide up just a little bit longer so that if you all have questions, you are welcome to reach out to Dr. Borman. And I would encourage you to do so, especially as you're learning. This webinar will be especially helpful to those of you who are working on horse quiz bowl and or hippology contests coming up, and also just good general information as we learn more about our horse project and we get sparked in our interests in 4-H. So once again, thank you for joining us. If you have any questions, be sure to reach out to Dr. Borman or to myself, and we thank you again for joining us. Okay.